Thank you, everybody, for coming today. Okay, so today I'm going to be talking about my thesis proposal titled From Glacial Cycles to Adaptation and Resilience, Linking Evolutionary History and Climate-Relevant Traits in Parnassius Butterflies. So we know that paleoclimatic events such as glacial cycles have driven both large-scale ecological community turnovers and changes in actual geographical features, and thus have contributed greatly to biogeographical patterning and organism evolution and diversification. We know a fair bit about how paleoclimatic events have impacted vertebrate animals, including the famous examples of mammoths and, of course, humans. However, insects have gone mostly ignored in this respect, despite being much more tightly coupled with their environments due to them being small specialist ectotherms. And anthropogenic climate change is accelerating environmental transformations at unprecedented rates. And alpine and Arctic regions are actually warming several times faster than the global average. Under this situation, insects, especially cold-adapted ones, are under threat, not only because they're biologically finely tuned to extreme seasonality, but also because they often occupy the upper limits of habitable space and cannot shift their range further to track changing conditions. Sorry, I'll start the time. So against this backdrop, I want to use my thesis to answer the overarching question: how have paleoclimatic events shaped niche specialization, and how have climate-relevant traits evolved in cold-adapted insects? So chapter 1 is going to answer the first part of the question: what are the drivers of niche shifts and expansions in these cold specialist insects? And chapter 2 to 3 are going to answer the second part of how have functional traits evolved to mediate environmental variabilities in these cold habitats. So a bit of the introduction on the study genus. So Parnassius butterflies is a genus within the Papilionidae family, which is swallowtails. So they're usually represented by large showy butterflies, such as the birdwings, as you can see on the phylogeny. However, Parnassius itself is a strict cold, often extreme cold habitat specialist. And here is a picture of a biotope that has around 5 species of this genus of butterflies flying sympatrically. I took this picture 3 years ago in Tibet at an elevation of 18,000 feet. And this type of habitat also represents basically what holds the most diversity of this butterfly genus. So this is like the ancestral habitat type. And these butterflies have many unique adaptations not seen in other ones, including but not limited to extreme cold tolerance, in which some species, immature individuals, can actually withstand temperatures lower than -40 degrees Celsius. And females also tend to lay eggs not directly on host plants, despite all species being specialist herbivores. And they also weave cocoons and sometimes actually dig subterranean chambers for pupation. And these are traits, again, not seen in really any other butterflies. And their larvae carry out behavioral thermoregulation by basking under the sun, which enable them to forage and feed at very fast speeds, which then enable them to finish development in a short period of the window that allow them to develop in these extreme habitats. Okay, so looking at the diversity within the genus, we can see that most of this diversity is within the Qinghai Tibetan Plateau, QTP, and Central Asian mountains in habitats as such, the open alpine kind of desert habitat type. However, there are 2 subgenera, the nominate subgenus Parnassius in red and the Dryopa subgenus in green, that have actually made their ways out of the high elevation places in Asia. And the Parnassius subgenus over here still kind of retained this ancestral habitat type of open alpine habitats, but in other geographical ranges. Dryopa actually were able to adapt to lower elevation, more temperate forest openings and edge habitat types as such. Okay, so here is a topology of the Dryopa subgenus, which is again here the subgenus that has actually specialists within the forest habitat type. So chapter 1 is going to be focusing on a vicariant species pair within Dryopa, Parnassius clodius and Eversmani, which are actually the only two species of the Dryopa subgenus found in North America. So Parnassius clodius in orange is found in mostly western contiguous United States, a little bit into southern British Columbia and Alberta. Eversmani is found across Siberia into North America. And they actually do provide us with a range of different habitat niche specializations, as shown here on the map. So using this sister pair, I want to ask the question, what are the drivers and consequences of niche shifts in these butterflies? Okay, zooming in onto the species pair, here is a hypothetical. You can't actually see my mouse. Never mind. Here is a hypothetical topology for the major clades within the two species. So under the big question, I have three sub-questions. So first, I want to ask, how did the species and their populations diverge? And for this, I have a hypothesis that niche shift in a single Eversmani lineage enabled their colonization of tundra habitats, while the rest remained in isolated forest habitats. And the second question is, how does niche specialization influence populations across space and time? And I hypothesize that the expected population size changes are going to be tightly coupled with how the lineage's niche specialization interact with paleoclimatic events. And the third question is how ecologically flexible are these butterflies really? And for this, I have the hypothesis that a broader apparent climatic tolerance / flexibility is enabled in lineages specializing in forest habitats due to the buffering effects provided by such habitats compared to the open tundra habitat types. So from my current whole genome sequencing data set, I was able to estimate the divergence times between the two species to be 2.6 million years ago, and the divergence time between the forest lineage and tundra lineage in Eversmani here on the bottom to be 1 million years ago. So this is given that this hypothetical topology is supported, but the placement of the blue branch here of the tundra Eversmani could actually be varied. But as long as this clade remains monophyletic, the story I'm about to tell you should remain mostly unchanged. So with that disclaimer and the modern distribution of these different lineages and some morphological evidence in which some of the forest Eversmani lineage actually exhibit this ancestral white wing color, in which all Parnassius have this white wing color. The only exception is the tundra Eversmani, which has a yellow wing color. So with that evidence, I was able to hypothesize the following divergence history for the two species. Okay, so during the Pliocene, Earth was much warmer than now, and forest habitat extended all the way into the Arctic, sustaining a forest specialist ancestor for both species. And around 2.6 million years ago, cooling starts as we enter the Pleistocene, and forest habitats start to shift southward, with part of this ancestral lineage following the southward retracting forest into North America via the exposed Bering Land Bridge. And then allopatric speciation basically happens as the two groups are now separated by the uninhabitable Arctic. And around 1 million years ago, the Middle Pleistocene transition period starts with intensifying glacial cycles, and tundra habitats became even more dominant for longer periods of glacial times. And this may have pushed a group of Eversmani to adapt to tundra, as there is no geographical barrier between the Siberian forest and Siberian tundra habitats. However, in North America, Parnassius Clodius is effectively blocked from the large continuous stretches of tundra habitat by the ice sheets. Here you can see in white. So basically, this gives them no opportunity to access and thus adapt to this newly dominant habitat type. And around 21,000 years ago, this was the last glacial maximum. The ice sheets were at their greatest extent, and they basically closed the corridor that was in between them, forming cryptic refugium within the ice sheets. So in the north, we have the Mackenzie Mountains, and in the south, we have the hypothetical Pink Mountain cryptic refugium. And here is a modern day distribution map of the two species. And in North America, we can see that the range is still closely aligned with what we see during the last glacial maximum. So their ranges still are kind of in correlation with the extent of the ice sheets back then. And just play that next video. So here is a sampling map. And the green pins are populations that has already been obtained and sequenced, and the following slides are going to be based on these green pin populations only. The blue pins are populations that I have access to or have been obtained, but is waiting to be sequenced, hopefully at the beginning of next year. And the red pins are populations that hopefully I can get, but right now I actually do not know if I can get them with any certainty. So since I am doing whole genome sequencing, the quality of the samples need to be high. So sample collection has actually been done by myself, other hobbyist friends, gladly, in Russia as well, and Greg from University of Alaska Fairbanks. So that was very helpful for him. It's a shame he's not here right now, but there way. I'll be first looking at population structure for these populations. And the current result is showing that KH, which I circled in red here on both the admixture and PCA, KH consistently stands out from the rest of the North American Eversmani population, as you can see here. Oh, you can't see my mouse. Yeah, anyway, the population circled in red consistently stands out, and the populations in blue are not very differentiated from each other, both from these analyses and also FST. So this is evidence that the species has a greater than expected dispersibility, or the more likely case of a very continuous habitat during the last glacial period. And as you can see here in this reconstruction, where the Mackenzie Mountains were isolated over there in the ice sheets, the Beringia was actually more or less a continuous big tundra landscape available for these populations to occupy and be basically continuous with each other. And to assess phylogenetic relationships, I'm going to be reconstructing species/population trees with different populations or population clusters if the interpopulation differentiation is very tiny or basically non-existing. I will also be looking for introgression evidence between populations. For example, on the right here, this is a tree mix graph from the current data set. And we do see that KH is, again, its own cluster sister to the rest of the North American Eversmani. And we do also see evidence for introgression between this population and the North Slope Alaskan population. However, because of limited sampling, I will not be further interpreting this result at the moment. Genetic diversity among population is going to be quantified using multiple indices. And right now we can see that both heterozygosity and nucleotide diversity are the highest in the Siberian forest eversmanni population on top, which again agrees with the hypothesis that this population is among the most ancestral by stable persistence in the ancestral geographical range, which is Siberia, and also a conserved niche specialization, which is forest. And inbreeding as another measure for genetic diversity is going to be estimated from runs of homozygosity and inbreeding coefficient. So again, the current data set is showing the lowest inbreeding in Siberian forest eversmanni. We can see over there the lowest, and also for the inbreeding coefficient is also the lowest. However, for runs of homozygosity, we see there is a mismatch in the Clodius sample between its runs of homozygosity. It has a very high long runs of homozygosity compared to its other indices for genetic diversity. So this may actually be due to admixture between a relatively inbred ancestral population and a more diverse population, as this sample population here does sit in the hypothetical context zone between two Clodius genetic clusters. And for cluster 1, it actually sits on the southern edge of the coastal distribution of this genetic cluster. For reconstructing effective population size trajectories, I will be using PSMC and moments. So using both methods together with previous structure and phylogenetic analyses would give me a chance to cross-validate demographic trends, divergence times, and also introgression. So here on the PSMC, I marked the different lineages in the different colors. So for green is forest eversmanni and blue is tundra eversmanni. And in the moments reconstruction, the curve on top is the North American tundra eversmanni populations, and the curve at the bottom is the forest eversmanni population. Also a little note, so for time progression for the PSMC, the present time is on the left, and for the moments on the bottom, the present time is on the right. So they're in opposite directions. So we actually see an expansion in tundra eversmanni using both methods around the Middle Pleistocene transition period around 1 million years ago. And in PSMC specifically, we see that there is an expansion in forest eversmanni effective population size around the last interglacial, which is 100,000 years ago. But for moments, we see a slow continuous inflation in the forest eversmanni as we get closer to present. So this actually overall does reflect the predictive trend of forest lineage tracking warmer times, whereas the tundra lineages are tracking colder times. I will also try to detect genomic signals associated with tundra versus forest niches. So I'm going to be calculating differentiation statistics for 3 contrasts between tundra and forest eversmanni, between Clodius and forest eversmanni, and Clodius versus tundra eversmanni. So I'm going to be identifying tundra shift regions where tundra eversmanni is highly diverged from both Clodius and forest eversmanni, while the Clodius and forest eversmanni remain relatively similar because they're both forest associated. So these candidate regions are then going to be examined with annotations to identify potential functions related to, but not limited to, for example, pigmentation, thermal tolerance, and immune functions. For the final component of this chapter, which was adapted from an original independent chapter that aimed to do niche quantification for the entire Dryopa subgenus. However, due to limited funding and diversity of ecotypes within multiple species within that clade, I have decided to narrow down to analyses to just this species pair, Clodius and Arsmeni, trying to cover all major populations and ecotypes. But again, fortunately, as you can see here, this species pair does still offer a diversity of niche specializations to be looked at. So I'm going to be reconstructing or estimating niche breadths from 3 dimensions. We have climatic, biome type, and habitat structure. So climatic niche breadth is going to be quantified as how much climatic PCA space each lineage takes up in the total space taken up by all lineages. While biome type breadth is going to be the effective number of biomes used by each lineage, which basically takes into account how many records are within each distinct biome categorization occupied by that lineage. So the plots you are seeing here is actually from my original independent chapter, which calculated these breadths on the species level. So we see that Parnassius Clodius actually have the highest climatic breadth, but the lowest niche breadth among all the species examined. And since habitat structure has been highlighted as an important factor contributing to Parnassius habitat preference, I will also be doing structural breadth analyses. So I will quantify the individual and joint contributions of both canopy height and canopy cover for structure. So here on the slide is canopy cover analysis again on the species level. And we did see the expected higher canopy cover in Parnassius Clodius, as it is a forest specialist. So to conclude the chapter, I will be inferring the ancestral states of the niche dimensions examined. This is going to give me another inference point for contextualizing diversification and niche specializations. I will detect phylogenetic signals for niche dimensions, which is going to inform me how ecologically flexible or conserved the different lineages and species are. And to evaluate the more macroecological implications of these niche dynamics, I'm going to be testing whether if the different niche breadths are correlated with each other. So positive correlations may suggest integrated ecological flexibility, but decoupled patterns may suggest more complex interactions between these different niche dimensions. So here is a correlation between the climatic and the biome type niche breadths on the species level again. So there is actually a non-significant negative correlation. And it is worth noting that the species on the bottom right corner, where it's the lowest biome type niche breadth and highest climatic niche breadth, are species that are strictly forest specialists. So this observed pattern here actually prompted the more or less post hoc hypothesis 3 for this chapter, which predicts that the possibility of forest buffering resulting in higher apparent climatic niche breadth for these species in the forest. And this pattern is actually going to be explored a little more in the final chapter as well. Okay, so for chapter 2, I will be looking at wing melanism in Parnassius. So melanism in insects is known to be associated with a range of biotic and abiotic factors, including predation pressure, desiccation, immunological defense, and thermal regulation. In Parnassius butterflies, wing melanism have actually been looked at in several species. Despite these studies all basically concluding there are complex roles played by melanism in these butterflies, the drivers and fitness consequences of melanism have not actually been tested experimentally in controlled laboratory or field settings. And the potential contributions of evolutionary history / genomic components of melanism have not been examined either. So for chapter 2, I will be trying to answer the question, what roles do genotype environment and genotype by environment interactions play in Parnassius wing melanism? I will be exploring this chapter using Parnassius Clodius as the most accessible variable and breedable species here in North America. And Clodius also offered me a population in the Gallatin National Forest in Montana that possesses a full spectrum of melanism levels sympatrically occurring. And this is not seen in any other population or species here in North America. So this population you see here on the right is going to be referred to as the focal population in the following slides. And a little note to make this a little bit clearer to people who's not familiar with this genus is that the default wing color of Parnassius is, again, white, and melanistic forms may appear, but no one has ever recorded them appearing without a melanism spectrum. So other Parnassius species, including Parnassius delphius from China, also have certain populations that possesses this melanism spectrum. So this thing is not really a singleton event seen in Parnassius clodius. Okay, so for the main question, there are again 3 sub-questions to be answered. First, I want to ask what are the environmental drivers of melanism in these butterflies? And I predict that melanism level is negatively correlated with temperature and solar radiation, but positively correlated with humidity. The second question is that whether if there is genotype by environment interactions, in which I predict that the focal population have variants that enable heightened melanin production compared to other populations. And finally, I want to ask, why does the focal population possess such a melanism spectrum, in which I predict that enabler variants are selected for in the population due to some anomalous environmental factors in its range. For example, a possibility could be that the habitat may be especially heterogeneous, either temporally or spatially.
Hey, Gary,
Yes,
would you like me to ask question now or later?
I think I would prefer finishing it first because the time could be a little tight.
Okay, go ahead.
Okay, thanks. Okay, I will be sampling populations not covered previously by study by Zaman et al. 2019, in which the authors explored correlations between environmental variables and wing patterns, including melanism in Parnassius clodius. They also did a bit of population genetics for the species using RADseq data. However, the study had a heavy focus in the Pacific Northwest region with pretty poor coverage in the in California and the Mountain West region, which includes the focal population. So for my whole genome sequencing samples, the population genetics analysis are going to be covered in chapter 1 together with Erismani as discussed. Wing melanism is going to be scored for all samples as melanism percentage on all 4 wings. And climatic variables are going to be analyzed with wing melanism scores to see if the previously identified trend of melanism being negatively correlated with solar radiation and precipitation can be observed as well using my data set. And this is also going to give me a look of whether if the environmental variables for the focal population habitat is anomalous in some way. Okay, so with the whole genome sequencing dataset and wing melanism scores, I will first carry out a GWAS within the focal population using melanism score as a response variable. However, hits will most likely not be identified because this population is likely very bottlenecked. In the likely case where no hits are identified, I will proceed to do genomic differentiation scans, similar to what I did in chapter 1 for niche associations, but with melanism as contrast. So I will be contrasting focal population versus a very closely related normal population, closely related population versus a distantly related population, and the focal population versus the distantly related population. So again, candidates are going to be identified as those that are highly differentiated between the focal versus the normal populations, both distantly related and closely related, but simultaneously not very differentiated between the normal populations examined. And to directly assess how environmental factors interact with melanism levels, I will be carrying out varying assays. So there will be a factorial design of temperature by relative humidity by solar radiation treatments for varying larvae from multiple populations, including a focal one. So my prediction is that melanism spectrum in the focal population may be a result from heightened response to environmental variables compared to other populations. As such, there are two types of outcomes. So the first one is the steepened reaction norm in the focal population, where the same set of environmental variables will induce more dramatic melanin production changes in the focal population than the other normal ones. And the second scenario is an extended reaction norm in which melanin production in the focal population is not capped or limited. I will also carry out reciprocal crosses between the focal population and a closely related normal population. So one quirk about Parnassius clodius as a species that makes it pretty nice for this purpose is that active males will readily copulate with females or unmated females if you place him upon her. So selective breeding is actually very easy in this species, which you can't really say that for most butterfly species. So if segregation appears in the offsprings, they are going to be sequenced, and the candidates identified previously are going to be further validated by melanism expressions in these offsprings. I will also be looking for signals of selection in these now more narrowed down candidates. And if we do not see any signals of selection, then melanism spectrum could be caused by neutral drift, which would also be surprising to see, as something so phenotypically significant would confer little or non-significant fitness costs or benefits. Okay. So I will also be directly assessing fitness, the fitness consequences of melanism levels. So larval fitness is going to be assessed using mortality, developmental time, and pupil mass under the same set of variables mentioned previously in the genotype by environment assay. And adult fitness is going to be assessed using female egg number and number of mates males have throughout their lifetimes. But the only environmental variable would be air temperature control within environmental chambers for these adult assays. So if I do see significant interaction between melanism and environment on fitness output, together with the previous habitat environmental factor analyses, I will be able to have a more concrete conclusion on the drivers of melanism in Parnassius clodius in general, and what is selecting for the melanism spectrum in the focal population. Okay, so moving on to the final chapter, I will be looking at cold tolerance and spring phenology in these butterflies. So overwintering, cold tolerance, and timing of spring emergence are very critical life history traits for cold-adapted insects. And under anthropogenic climate change, both more frequent winter warm and cold extremes and reduced winter snowpack can threaten overwintering for survival in these insects. Early spring warming and reduced snowmelt may also disrupt spring phenology, compromising development the next year. Despite this, studies regarding these highly sensitive early stage traits are lacking in cold specialist systems. In Parnassius, overwintering mainly occur as a quarantine enclosed parade larva, which I will here on after refer to as just overwintering eggs. And within the genus, again, we see a range of different cold habitat specializations varying in snowpack, temperature, and host plant biology. So for this final chapter, I want to know what drives variation in overwinter cold tolerance and early spring phenology in these butterflies. So again, there are three sub-questions. First, I want to ask, is variation in cold tolerance from local adaptation, plasticity, or both? And I predict that cold tolerance is both locally adapted to snowpack and show plasticity to environmental conditions experienced. Second, I want to see if supercooling point, SCP, is a valid proxy for cold tolerance. And I predict that it is valid, and it's negatively correlated with overwintering survival. And thirdly, I want to know if there is local adaptation in spring hatching queues. And the hypothesis is that hatching queues are locally adapted to both the spring local climate and the host plant biology at the habitat. Okay, so first, I will be examining cold tolerance in 5 Parnassius species across a holarctic distribution covering various habitat types. Supercooling point will be measured using eggs by cooling them at a constant rate in the freezer as a setup here you see on the top right. So far, I have gotten results from 3 species with the sample population shown here on the map. So in the forest specialist Clodius here, you can see in the green circle or square, I saw a clear trend of significantly increasing SCP, which represents a decrease in cold tolerance with increasing elevation and snow cover, which is likely due to the fact that snow effectively decouples under snow temperatures from that of the actual air temperature above snow, resulting in a warmer, more stable environment for these eggs that are overwintering at higher elevations. However, for the Parnassius namian and the 2 populations of Smintheus here on the right, which are temperate and subarctic grassland specialists, they all show a pretty conserved, lower, and not significantly different SCP. So despite all of these different populations experiencing different winter temperatures and snowpack volume in their respective habitats. If we look back at the niche estimations in chapter 1, we can actually see that this is somewhat reflected in the climatic niche breadth of these species. And as we see, the Clodius have the highest breadth, while the 2 other species have consistently low climatic breadths. And with the SCP pattern observed here, there is a possibility of a relaxed selection in realized thermal tolerance in Parnassius Clodius, which again inhabits forests, where these local habitats or populations are allowed to have their own thermal optima due to the buffering effects provided by forests. Whereas in the other more exposed treeless habitats of these other 2 species, strong selecting pressure may be exerted by the very harsh unbuffered winter air temperatures, which basically canalizes their winter cold tolerance to be very conserved and low. I will use overwintering mortality assays to validate SCP as a proxy for winter cold tolerance. So for this experiment, I will only be using 2 species, Clodius and Smintheus, as I can only obtain large enough quantities of eggs to run these kind of assays for only these 2 species. So there are going to be 2 acclimation temperatures and 2 overwintering temperatures. And SCP measurements are going to be carried out on a random subset of the eggs, while the rest are allowed to hatch the next spring. And this is a prediction heat map for how overwintering treatment temperatures and SCP are going to interact to determine mortality. I also do predict that acclimation temperature will play a role, in which high acclimation temperature may lead to increased mortality under the low overwintering temperature due to its failure in prompting the production of cryoprotectants. Okay, so to validate the lab results, I will also be carrying out field mortality assays. However, this is going to be done only on Clodius, which has accessible habitats through a wide range of elevations here in California. So populations at different elevations or different habitats are going to be reciprocally transplanted, and spring queues are going to be measured. So 3 OE position sites are going to be chosen at each habitat by following females the previous summer, and eggs are going to be overwintered in mesh bags secured at these sites, together with I-button loggers for in situ temperature logging. So if there is local adaptation in SCP, and that SCP does reflect winter cold tolerance, I am expecting to see heightened mortality in both the low and high elevation populations at the mid elevation habitat, which represents basically a turning point where winter temperature is low, yet snowpack is not as stable as the high elevation habitat, making it effectively the habitat with the lowest experienced temperature for the overwintering eggs. However, there could also be local adaptation in metabolic cost regulation, in which case we may also see heightened mortality in the mid and high elevation populations at the low elevation habitat, as these populations may have more limited capability to suppress metabolism under warmer winter temperatures. For the final component of this chapter, I will be looking at spring hatching cues in Parnassius Clodius. So these cues and how the larvae respond to them is especially critical in systems where developmental windows are short and host plant availability is limited. Premature hatching may risk larval mortality from starvation or low temperatures, while delayed hatching may cause larvae to miss the brief window of host availability. So across elevations, Parnassius Clodius feed on hosts with very different growing schemes. At lower elevations, Dicentra formosa is used, and this species grows to relatively large sizes that last well into midsummer, providing the larva with both an abundance of food and longer availability window. Whereas at high elevations, Dicentra passiflora and uniflora are used, and these plants are spring ephemerals that grow basically very few leaves before flowering and then senescing, providing larvae with less food abundancy and shorter availability windows. As such, populations at higher elevation may have increased Q sensitivity due to selective pressures imposed by shorter developmental window attributed to both the local climate and host plant biology. This is actually supported by this analysis I did on adult flight span at different elevations. So this bar graph is based on GBIF records of Parnassius Clodius. We actually do see that higher elevation bands here in blue consistently exhibit shorter flight periods, supporting the hypothesis of a stronger phenological constraint at higher elevations. So I predict that low elevation population is in warmer habitats may require greater degree day accumulation before hatching, basically buffering them against false springs, which can be frequently experienced in such habitats. Whereas high elevation populations using ephemeral hosts may hatch with less degree day accumulation and be more sensitive to humidity increases from a snowmelt, promoting synchrony with their host plant biology. So to test these predictions, overwintered eggs from populations spanning elevations and host types are going to be exposed to temperature by humidity treatments, maintained using environmental chambers for temperatures, and a 48 well plate design for humidity as shown here. So in this design, humidity levels will be controlled for each egg independently, reducing pseudo-replicates. So time until hatching will be recorded and developmental zeros and degree day requirements are going to be estimated for the populations. And on the right here is data from last year where we do see shortening of time until emergence consistently in the wet treatments. In this is actually a mid-elevation population that feeds on the bigger host, Dicentra formosa. For higher elevation populations, I am expecting to see shortened time until emergence overall, with the wet treatment also further shifted along the way. Okay, so together, I hope these three chapters are going to establish a framework linking evolutionary history and functional traits in these cold-adapted Parnassius butterflies. And hopefully some of these patterns observed here could be generalized to other cold specialist systems, which are very much under-investigated this. Okay, that's it. Thank you all for listening. And now I'm open to questions and suggestions.
Hey, Gary, you haven't actually write the thesis proposal yet, right?
I did.
And because did you send it to us?
No, I didn't know
Oh,
I had to send it to you. I thought it's only for the QE committee.
okay. Yeah, no, you've already written it. Like, it'll be great if you can send it.
Okay,
Yeah.
okay,
Um,
I'll send it to
so for chapter one, you're gonna do a lot of sequencing, and then, for chapter two and three, especially three, you're gonna do a lot of like some functional analysis.
you.
Are you going to keep some of the samples that, maybe freeze them or something like that after you score the phenotype? You
Yes,
know, you
for
might,
like melanism level.
yeah, you might be able
Yes.
to, once you get, because you're doing two and three sort of separately, independently in parallel from one, where you already have some sequencing data, but you might be able to, if you have frozen samples from two and three, once you get and analyze and finish analyzing some of the sequencing data and point to sort of some of the, players that are involved, you might able to, be able to score them
Get some
and
signals
yeah,
from the yes.
yeah.
Uh-huh. Yeah, that is the plan. Yeah.
Okay, great.
A question about I guess with the focal population where you have all this variation in the melanism.
Uh-huh.
Well, I guess several questions. The first one is, I mean, you kind of alluded to the possibility of there being some sort of adaptation or some sort of selective pressure that is driving the
Right.
melanization.
Mhm.
But I guess one possibility could be that this is like a genetic drift or some other
Yeah, it could be
factor
from
affecting.
drift.
I mean, it's hard to, I guess, disentangle both of them. But I get, yeah, maybe you could say something about how.
I'm gonna try to do selection scans on the genomic regions identified. I don't know how. I've never done that before, so I don't know how easy that that can be done. But it looks like I can do a lot of tests to see whether if it would be caused by drift. And I'm also thinking if I do have enough resources, I want to sequence the other Asian populations that also have this melanism spectrum to see whether if there is some sort of pattern. For example, are all these like melanism spectrum populations extremely isolated and have low genetic diversity, which could support more of the drift possibility. Yeah.
Well, and the other, it's a more technical, well, question and comment is, well, you're doing a lot of breeding experiments and sequencing to try to identify, with FST, DXY, all these different metrics to identify the candidate regions. With that is exactly what I would do. But in addition to that, you might consider doing, if you could breed those populations, so you could at least rear them in the lab. You could do differential gene expression in developing wings. I guess if you knew ahead of time, this is going to be a melanic, this is going to be a non-melanic, you could take the extremes and then take a few samples of each and do transcriptome sequencing, RNA sequencing, and that could help you narrow down candidate genes.
Is RNA-seq like expensive?
No,
Okay,
it's very cheap now.
that's
I
good.
mean, everything is relative, but you can get a really good data set for I don't know, like $3,000, something like that.
Great. Well, yeah, I'll definitely consider that if my resources are not limited. Because right now, I feel like all my funding is going into the population genetics part of this right now with my limited funding, and that's struggling to cover everything. But
when I qualify that comment by me, yeah, cautious. You're doing several PhD theses there, so I don't want to suggest this is like, oh, you should do this because it's already a lot. But a possibility.
yeah.
I mean, I guess depending on the data that you get out of the population genetic sequencing, if you find already really strong candidates, the best way to validate those would be with the RNA.
RNA-seq, right?
But you may think about it as you get some of the data.
We'll see.
Yeah.
We'll see what.
Gary, do the melanic butterflies have a lower scale density? It seemed to be.
I do not know. I also don't know if I can measure that. I haven't looked into it. They look transparent.
They look more
Yeah.
transparent.
Yeah. But that's a good point. I should look into whether if I can do it. But I also don't know if they do have a lower scale density, what I'm gonna make out of that.
You could presumably just count it scale density under a scope.
Yeah, but what am I gonna make out of that data?
Well, I guess one question would be whether they're actually melanic or
Just lacking scale.
lack scales.
From what I see, they have scales on those parts that you see as transparent. I mean, Parnassius is in general pretty transparent, but like you can see here, even the white part you can see through to the little grids or the mesh I used underneath it. So we'll see. That's a good idea. Maybe I can do like a preliminary pilot just by zooming in and counting scales.
What is your thinking behind your hypothesis that humidity has the strongest, is like a predictor?
So from a previous study, they concluded that solar radiation and precipitation had the strongest effect on wing melanism. So one possibility is that why I'm thinking about humidity is that humidity could confer some sort of like increased risk of infections for these butterflies. And we know that melanin is some sort of important in immune function. So that could be one possibility. And also from anecdotal experience from a hobbyist friend of mine, they're saying that apparently for another species in China, the melanic forms are seen more frequently in the more humid habitat types. So, but that's only anecdotal. Like it could be just observer bias. Like he thinks something is acting on it, but it could just be neutral. We don't know yet.
I was just kind of curious for maybe like selection pressures on wing melanism with like predation of these butterflies influence that, or like is there a major predator?
It could influence. I know the previous analysis didn't identify any prediction pressure, but my analysis is also not going to address that prediction pressure. But if I do get signals of selection for melanism, and I don't get any signals from like other, I guess, more or less abiotic environmental factors, I will be looking at potential predation pressures. Okay. Well, it is 3. And if there's no more suggestions or comments, I guess we're over here. Yeah. Well, thank you to the folks on the Zoom as well.